हेलो। हेलो। हेलो, माइक चेक। हेलो। 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 हेलो।
हाँ ओके ठीक है हाँ
माइक चालू था, सब अच्छा आवाज आ रहा है। नहीं नेट तक ही नहीं लगती।
हेलो, माइक चेक।
क्यों? नहीं नहीं पूछा देखते हैं। भेज देना एक है ना इसको तो। नहीं वहाँ से सिलेक्ट कर, वहाँ से सिलेक्ट कर। केबल हिल गया।
माइक चेक 1 2 3। माइक चेक 1 2 3।
वो माइक चालू है क्या? हाँ हाँ जी, वो माइक चालू है क्या?
हाँ चालू है।
बंद करो ना, खाली पूरा आवाज़ जा रहा है।
कौन सा उधर का?
हाँ
हम बोल रहे तो आवाज़ जा रहा था।
हाँ। कहाँ से आवाज़ आ रहा है इतना? आवाज़ कहाँ से आ रहा है?
एम्बियंस आ रहा है क्या?
हाँ
एम्बियंस आ रहा है?
नहीं, इतने लोगों का आवाज़ कैसे क्या आ रहा है? नहीं आना चाहिए ना।
हेलो 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 हेलो
किधर था कन्वर्टर?
हेलो हेलो
Hello. Shunu. Good afternoon. Welcome you all for the Science Day celebration. Uh, yes, can you just settle down? Whoever is coming in can fill in the back seats. So I'll first welcome our director, Dr. Ashish Lele, to give his welcome remarks. Ashish.
Why don't you, all of you guys, come in the front, please? I'm talking to everybody sitting there in the last 10 rows. Please come. There are many vacant seats here. Please come. Okay, all of you students, just remember this. At some point of time, you are going to come and stand on a podium like this. You are going to face the audience, and it doesn't look good for you. When you come and stand here, you will not feel very good if you see empty seats. You understand what I'm saying? Please imagine yourself in that position. When you come and stand on the dais and many seats are empty in front of you, you won't feel good. So I'm requesting all of you to please occupy seats at the front. And let this be a standard way. I don't want to tell this to you every time I come here. Okay, good afternoon. Today, as every year, we are celebrating the National Science Day, February 28th, 1928 was when Sir C.V. Raman discovered the Raman effect. And this is particularly for students. I don't know how many of you have read this very small booklet that was printed by the IACS in Kolkata in collaboration with the American Chemical Society. It's a very small booklet. It tells you in very succinct words the history of Raman effects, and it also has a small biography of Sir C.V. Raman. There is also a very wonderful biography of Sir C.V. Raman. I believe we have a copy in the library, and those of you who want to be inspired by what Raman achieved back in those days, this is a wonderful biography that I will encourage all of you to read. So, like I said, February 28th was a landmark day when Raman discovered the fact that when light passes through a gas or a solid or a liquid, it gets scattered. Most of the photons that go through the medium get scattered as per the standard Rayleigh scattering, but a small fraction of those photons change wavelength, right? And this is basically what he discovered. It was first announced on March 16, 1928 at the South Indian Science Association meeting in Bangalore. And almost immediately after that, on the 31st of March, 1931, in Nature, volume 121, page 501, 502, a two-page paper. I think it was titled A New Type of Secondary Radiation. And that's when the Raman effect was announced to the world. I think the rest is history, as they say, and it was first the physicist who got enamored by the Raman effect, because it was one of those experiments that that supported the quantum theory at that point of time. So the physicists got really excited about it, and about 700 papers came up very quickly trying to understand the fundamentals of Raman effect and the interaction of light with matter. And then the chemists took over because it was found that every material had a certain specific Raman signature. So it became a tool to characterize materials. And of course, it really came into fore with the advent of more sophisticated computing power and particularly with laser technology. So Raman effect really came to fore after that. So I really urge you to read this wonderful booklet. So in celebration of that fascinating discovery, very fundamental in nature, India has been celebrating the National Science Day every year on the 20th of February. It's very nice to see that we are participating in that celebration, and it has been wonderful to see about 170, I'm told, posters which have been put up in the main building. And that's the display of all the nice science that all of you guys are doing. So it's very nice to see the excitement with which you are participating in this wonderful celebration. I think there was a jury that went around and wanted to decide which of those posters deserve a recognition. I'm sure the jury had a tough time going through that, and I think there are 30 posters that were selected for awards. So with that, I just want to welcome all of you today afternoon for this celebration. We are going to have a nice event, a wonderful lecture by somebody who's eminently suited for this occasion today. He loves this interaction between light and matter at the broadest level. So a wonderful lecture follows, and then there will be a short session on the award presentations and recognizing the quality of science that we are all doing at NCL. So with that, I hand over to Sarika.
Thanks, Ashish. Yes, as you rightly said, I guess Arindam is the right person to speak today because to celebrate Raman, C.V. Raman, I guess he's the right speaker. I'll request Shayan to formally introduce our today's chief guest.
Thanks, Sarika. So it gives me immense pleasure to introduce today's speaker, Professor Arindam Chaudhary from IIT Bombay. So he obtained his bachelor's degree from University of Calcutta and his master's degree from IIT Kanpur. Then he did his PhD from Carnegie Mellon University, and after that he had a postdoctoral stint at Columbia University, and then he came back to India and joined IIT Bombay in 2006, where he established one of the first single molecule fluorescence spectroscopy lab in India. So his research focus on his research focus has been to understand spatio-temporal heterogeneity in soft materials, semiconductor nanocrystals, and perovskite crystals. So he is also involved in the development of imaging methodologies and sensing analytes in biological media. So Professor Chaudhary is affiliated with the National Center for Photovoltaic Research and Education and the Wadhwani Research Center for Bioengineering at IIT Bombay. So this was the formal introduction, and I want to add a note just for today, because Arindam and I were there at a conference, and exactly 7 days back, and this conference was of the chalkboard format. So when Arindam was giving his chalkboard talk, the chalkboard flipped and fell on his toe, which fractured his toe. And in the last 4 days, he had a couple of microsurgeries. So and we are actually very grateful to him that he is here today, although the last microsurgery was yesterday evening. So with this, I invite Arindam to start his presentation. The stage is yours.
Thanks, Sayan, for the introduction and telling everyone else about the discussions we had yesterday. But I intended to come here for this special occasion. And can you hear me? Can you hear me? Yeah. And it's an honor to be here for this very special lecture on Science Day. And I again thank organizers and especially Ashish, Sarika and Sayan and the team who decided to call me. It's really good to see. Okay, all right. I need a pointer. If you guys have a pointer, that will be nice. Be careful. That's fine. That's fine. Okay, that's fine.
Off. Yeah, it's off now.
It's off, right? Okay, good. So pardon me, I am going to be limping a little bit. No, I'm fine. I'm fine. I'm fine. So let me first tell you that I do basic science. I do fundamental science. I don't really think too much about applications, and I'm sure even Raman, when he discovered the Raman effect, or for that matter, any of those guys back in the early 2000s or 1900, whatever, when they discovered things, they probably did not think about what applications will happen in the future. And we all know that how Raman effect, how much applications it has and how much useful it has become for chemists, material scientists, and even engineers, right? So for me, my philosophy in science has been to look at fundamental processes. And today I'm going to discuss with you our journey over the last 8 years or so on these crystals of different, some of them nanocrystals, some of them bigger crystals, of this crystal called perovskite crystals, which many of you have heard about. And so I'm going to tell you about one of our discoveries, which probably contributed a little bit to the knowledge of optical properties or optical instabilities in these perovskite crystals. As you can see, I represent the single emitter spectroscopy group at IIT Bombay. Okay, in my lab, very briefly, we are interested to look at the spectroscopy and the dynamics. We do spectroscopy and dynamics of single emitters. And by single emitters, essentially I mean single molecules or single nanocrystals, anything that emits light, I call it an emitter. And over the years, I have been working on semiconductor nanocrystals or quantum dots or quantum confined structures. Many of you have heard about quantum dots and quantum rods. More recently, I have been involved in this materials or these crystals, which are perovskite crystals, which have a lot of potential applications as solar photovoltaic materials as well as light emitting devices or light emitting diodes. Apart from that, I have also been working, although that part of that soft side in my group, this I call the hard side, and this is the soft side, has shrunk a little bit, trying to understand dynamics in soft materials, okay, including polymers. And if I some of you may have been there, I gave a talk about 7-8 years back on this aspect. And more recently, I have been involved in development of methods for fluorescence-based imaging and sensing in biological cells. But apart from developing such tools on methods and analytical methods on even on measurement and analysis, the main goal of our lab is to essentially probe the degree of non-uniformity of a physical property And that non-uniformity can be in either space or it can be in time. And that's what I call as spatial or spatiotemporal heterogeneity. Okay, that is really the focus, because conventionally, if you think of an ensemble experiment that we typically do, so just simply taking a measurement of an absorption spectra or emission spectra in a cuvette, what you're doing is you are collecting data which is averaged over millions of molecules. And what you get is an average number. And that's great. That's great if you are looking at an environment which is homogeneous, like say water or a pure solvent. But whenever you go from that environment to a situation like in a biological cell or even in a polymer matrix, the environment keeps changing, then such an average is not enough. We need to extract more information, and one of the ways to do it is to probe the sub-ensemble. Ensemble is the entire thing. To probe the sub-ensemble, I'm giving an example here. Imagine I have these green, orange, pink, and blue balls. These balls represent this environment which are different from each other for certain properties, and you may have some dye molecules or something else which are kept in these balls, and they will have a different response. So they can be different polarity or different rigidity or whatever it may be. And you may get an average spectra, for instance, which would look like this. Please focus on the left panel. If you take the data from this entire ensemble. But if you instead, if you start going to collect, and if you go and collect data from smaller and smaller sub-ensembles, and of course single molecules are essentially the ultimate sub-ensemble where the molecular property is still retained. If you break the molecular properties change. Then you'll see that different kinds of spectra, for instance, can be obtained, and the sum of all these things would end up getting what you have in the ensemble spectra. So what's the real advantage here is that you can construct distributions of physical parameters, okay? And once you have distributions of physical parameters, not only can you get the mean value or the average value, you also know the width of the distribution, and the width of the distribution essentially is a indicator for the extent of heterogeneity in the system. This particular example that I'm showing here is not a function of time but a function of space, but you can do exactly the same thing as a function of time as well, how things change with time. For instance, how the intensity fluctuates with time, and that is actually going to be a focus of today's talk. So this variation in space and time is what we are interested in, and in this process of detecting single molecules or even going to the sub-ensemble, we can essentially unveil or uncover many rare events which we would not be able to obtain in a bulk experiment, okay? Because it's obscured by the averaging effect, and we can obtain insights on mechanisms, and more importantly, we can also discover some new phenomena. And I'm going to give you an example of that, a few of them today. Now, I'm not going to go into the details here, but this is a setup, an optical microscopy, fluorescence microscopy setup that we have built in our lab. It's actually reasonably cheap. If I wanted to buy it, it would probably cost me about 2 to 3 crores, but we can take different components and we can build this up in our lab, and we can do all the measurements, and we have way more flexibility than a commercial machine. Approximately 1/4 of that money, all right? And this flexibility gives us opportunity also to do various kinds of measurements. We can look at individual molecules or particles which emit light, as you can see over here. We can look at diffusion dynamics. If these things are moving around on a surface, we can probe them and understand the property of the matrix where they are in. And also we can collect emission spectra. As you can see, these individual streaks that you see, these are the emission spectra of individual particles. And we can not only resolve them in space, but as you can see here, we can also collect a video or a movie of the data, and we can get the information of how things change over time, okay? And that is really how we do our measurements. Not going into too much details here. Now, what's the plan for today? The plan for today is to give you an idea, some background on what we call as blinking. Some of you have heard about it. That is another name of emission intermittency, and a few examples of blinking and flickering that I'm going to talk to you about. Remember, I changed the title of the talk a little bit. It's wink, blink, flicker, and sparkle. So hopefully, I'll be able to give you an example of each of them in terms of optical instabilities. And then I'll be talking about a weird phenomena that we found out, kind of accidentally, and more importantly, why and how. Because as scientists, it's my fundamental job is to actually ask the question why something is happening or how something is happening, rather than just reporting a phenomenon. And if I have time, I will talk about something that we don't understand yet. Okay. So this is an example of standard molecules that all of you probably have seen. This is rhodamine dye, this is a cyanine dye. This is a dye aggregate. 4 perylene dyes connected to each other through like a central carbon atom. This is a green fluorescent protein. I'm sure many of you have heard about green fluorescent proteins or yellow or red fluorescent proteins. This is a semiconductor nanocrystal. It's a TEM image of a semiconductor nanocrystal. Dimension is in the order of about 3 to 4 nanometers. All right. This is an example or a cartoon picture of a conducting polymer. The huge applications of conducting polymers. And if you take any of these guys and if you dilute it, basically do homeopathy in a solvent, and then you cast the solvent out and cast it on a cover slip, which is very clean cover slip, you are going to see a spot which would look like this. And the dimension of this spot is typically in the order of 200 nanometers, and that's because that is the diffraction limit of light. So when you are using photons, I will always be restricted by the Abbe limit, which is the width of this distribution is in the order of few hundred nanometers. So compared to that, this central red square that I have shown here, within that you have a spot there that is a 10 times magnified image of what the actual size of the molecule is. So first, I would like to clarify that when we use the term single molecule imaging, we are actually not looking at the molecule. We look at the light that comes out of the molecule. Okay? This is something that I want to clarify. More interestingly, this is a static image. If I collect a movie as a function of time, if I keep collecting, I will see something like this. Light will come out at a certain point of time, it will stop coming out at a certain point of time, and this fluctuation will keep going on till the molecule is destroyed. And if I sit on top of this molecule, collect the data and plot it as a function of time, I will get something like this. And this is for a polymer, conducting polymer called MEHPPV. This effect of at arbitrary time points, fluctuations of intensity at arbitrary time points is called blinking. Okay? And you can see these sharp jumps. I'm going to come to that in a moment. But this is, this blinking process has been for a long time, last 25, 30 years, has been related to single molecules or single emitters. And often it is touted as being a bad thing, because if you want to use a single photon source, you wouldn't want to have light coming out at a random time and then no light coming out at a later time, right? So that's bad for that. Or even if you want to track a particle, if the particle disappears, then you don't know what you're looking at. That's bad. On the other hand, it can be good, because this blinking process has been used for what we know now as the super resolution imaging, where you can get down to nanometer scales in imaging in biological as well as in material samples. So this is the basic idea of blinking. And blinking is, as I told you, is ubiquitous for any molecule that emits light. And this is an example that I'm showing you of how a molecule blinks. All the spots that you see, they are immobilized molecules. All the spots that you see, they are immobilized nanocrystals, and they are in a rigid environment. They can't move around. And you can see there's a clear difference in their patterns, isn't it? This is a faster blinking going on, there's a slower blinking going on. And if I sit on one of the molecules or nanocrystals, you will see this kind of sharp jumps up and down. In nanocrystals, it's a little bit fuzzy. You can see there are a lot of states which are not clearly on or clearly off states, and this is typically what you would observe. But more importantly, this temporally random phenomena, when I say temporally random, that means I cannot a priori tell that, okay, at this instance, my light is going to turn off, or at that instance, my light is going to turn back again. That is unpredictable. That is completely not, it's random. This coupled with another important thing about blinking is that this abrupt or discrete jumps that you see between on bright or a dim off state. Okay? That is also important to know. Why is that? The reason is that if you think of a let's say talk about nanocrystals, when you shine light on nanocrystals, what you do is you generate an electron hole pair, and this bound electron hole pair is called an exciton. Now, what essentially in the language of chemists or molecules, you are essentially taking the system from a ground state to an excited state. You have an electron here, you have a hole here, and this electron can come back and recombine and light can come out. The electron and the hole is just a plus charge, can recombine and light can come out. And this process, if it keeps going on, then I would see a light on state or a bright state. But once in a while, it's a rare event, this electron can get trapped in one of the surface traps that are present in these very small nanocrystals. And if this trap is a long-lived state, then the ground state can no longer be excited, and therefore, even though you excite the system, no light comes out. Okay? And this phenomena has been explained in nanocrystals by the charging and the discharging of the core. Although this trajectory is for molecules, I am just giving you here as an illustration. Once a photon comes in and you generate an electron hole pair, the electron can get trapped on the surface and you have a plus charge here. Moment that plus charge exists, the light emission stops. When they recombine, light starts emitting again. And that's the reason why you see these very abrupt changes in the emission intensity, and that is a feature of blinking. Now let me switch gears and talk about a little bit about the system that I am going to talk to you about today, and that is this crystal called this perovskite crystal. And many of you have heard about it because of late they have been touted to be the material that we need for next generation photovoltaics as well as for light emitting devices applications. And one of the very important factors is this high carrier diffusion. By carrier, I essentially mean electrons or holes or electron hole pairs that are made in this that are there in the system once you shine light, and they have very high carrier diffusion lengths and diffusivities, and therefore what the idea is that these electrons and holes or these charge carriers, they can migrate large distances and they can be captured, and that's how you can convert photon energy or light energy to electrical energy. And also they have very high absorption coefficient and fluorescence efficiency as well as lifetime. And this is also a very important property which tells a little bit more about the defects that are present and so on. Another interesting thing about these systems is that they're chemically modifiable. For instance, here this is an all inorganic version. This is probably a cesium lead bromide, and this is a picture, this is a cartoon where I have replaced the cesium atom with a methyl ammonium atom, all right, molecule. And then that gives rise to another perovskite which is called the methyl ammonium lead bromide. But the point is that you can change the halides, or you can change the lead, or you can change cesium, you can change the system, you can change the color of the light that is being emitted. And that is the reason why these are touted to be used for light emitting device applications as well. Now, for our lab, our prime interest was to look at the optical instability of these materials, because when we started this project back in 2014, there was no prior study that was there on the behavior of these nanocrystals at a single particle levels. And we also wanted to look at the atmospheric stability, because we all know that if you want to make any of these devices, especially in places like Mumbai, where we have a huge amount of humidity that is present, and if your TV stops working, that's no good. So that's one of the reasons why we wanted to look at the photo, and photon means light based, that's for photovoltaics as well as the atmospheric stability. That was the real goal of when we started off. And in fact, in the very beginning, I worked with Anshuman Nag, and many of you heard about him. He is in CSIR Pune, and he made these beautiful crystals based on another work that was just published the same year. And we started looking at individual crystals, these cubic crystals of cesium lead bromide. And as you can see, the spectra is very narrow, very clean, but more importantly, they're very, very bright. I have never seen my 15, 20 years of experience in single molecule spectroscopy, yeah, almost 20 years. I have never seen brighter single particles. They are so bright that I can't even see the noise in the signal. Now, the funniest thing was that once we put the sample on a cover slip, it's a simple glass cover slip, we did not see any emission, which was a huge surprise. And this is exactly what we saw, and you will be surprised of what I am trying to tell you. This is where my cover slip is, and did you guys see what happened? I will go back again and I will replay this. Nothing, something's coming up. I am continuously shining light, and pop, 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 pop. One crystal of the other starts emitting light, and then it stabilizes. I stop light for some time and I redo the experiment and see what happens. After stopping the light and keeping 2 minutes in the dark, again they disappear, and then again they start re-emitting light, for the majority of them. And this told us about something, that this kind of popping up behavior must be something to do with oxygen as well as water molecules that are there in the atmosphere, which get absorbed on these crystals, and they can create defects. These defects are essentially the result in non-radiative processes which funnel all the carriers to recombine through non-radiative channels. And once we heat the nanocrystals, then these molecules can dissolve and these defects can disappear, and then you can see the emission coming off. Now, this image that I show you here is actually not an image. It's actually a movie. You can see that some of them are flickering. If I sit on top of some of them and plot the trajectory, you can see that for the most cases, the nanocrystals don't ever turn themselves off completely. They basically undergo something called flickering, as you would see a flickering of a light bulb or of any of the LED lights. Sometimes you see this very flickering. It doesn't completely turn itself off, but for a 10 to 15% of the crystals, you can see these complete off states existing, and these again are dependent on the oxygen and the moisture that is present around them. So the bottom line is that this process of flickering essentially owes to the fact that the lifetimes of these non-radiative states that are there, where the electron and the holes can get trapped, they are actually not very long. They are in the order of milliseconds, and therefore we see flickering and not completely a blinking behavior. And this also can explain the nature of these materials being tolerant to defects. That's another way of saying that these are tolerant to defects. But moisture definitely enhances this blinking to this off state, which is a clearly off state, by generating these defects that are present on the surface. Now, I'm going to go back to the picture that I had shown you before, where I talked about this electron and the hole being separated, and they are not going to be emissive. And this is the part where the electrons get trapped on the surface. And this is a picture of the energetic picture of the trap of the states. And the idea again is that once they get trapped, they cannot emit the electron in the hole. Once they come out of the trap and go back and merge with the hole in the crystal, you can shine light on it again, and the emission process restarts. So this also tells us about the importance of traps in these nanocrystals, and traps are responsible for this process. And so blinking conventionally has been attributed to nanocrystals which are quantum confined. What is the meaning of quantum confined? By that, all of you probably have heard about particle in a box model. You have a particle which is confined in a quantum particle confined in a potential, infinite potential well. Essentially what you can show is that as you constrain them, the energy levels change, energy levels increase. So you can think of it in a very, very similar fashion. So in situations where we have materials which are extended materials such as 1D, 2D or 3D, here I should not have or should not see the effects of the surface, because here the confinement is less, effects of surface are relatively less, and the most of the carrier recombination does not happen through the surface but happens through the bulk of the material. And other than that, in an extended object like a wire or a plate or a cube kind of a thing, which is a big crystal, you don't expect to see blinking phenomena. Why? Because you can imagine something happening here and here and here and here, or here, 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 here. They will undergo blinking locally, but if I take the whole thing together, there will be an ensemble averaging effect. That means I'll average things out and I will not be able to see these sharp jumps of on and off states that we would see in a nanocrystal. And that is really the conventional wisdom. So spatially temporally uncorrelated functions for fluctuations of the intensity should essentially cancel out. Now there have been few reports, and first report actually goes back earlier, but I'm giving you an example of these CDAC quantum wires. These are again extended objects, but they are still quantum confined systems. And if I play a movie, you will see what I mean here. You can see that there's intensity fluctuations happening, but they are spatially heterogeneous. They are not behaving the similar fashion as a single crystal. And this is another example of a 2D material. This is a reduced graphene oxide, and it is actually getting oxidized, and you can see these small itty-bitty fluctuations. And if I plot the intensity from a local area, this is from another paper, you can see these fluctuations on top of the overall background. That is also touted, or basically you can call it a blinking or a flickering, but again they are spatially uncorrelated. That means the fluctuations happening here, 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 they are different. On and off states are being accessed at different points in time. And this fluorescence or PL blinking or flickering essentially was not reported without any quantum confinement. This is again a quantum confined structure in this direction, not in this direction, in plane, but out of plane. Okay. Now, in 2015, my colleague Shaibal, he was trying to make these devices, and for these devices, he was looking at this methylammonium lead iodide films. And we started collaborating on these projects, and you can see that this is a SEM picture of these films of methylammonium lead iodide, and you can form these discs which are typically between 2 to 5 micron in size, and they are, and AFM picture tells you they are not quantum confined because they are quite thick. And once we put these samples on a glass cover slip, it's easy to make, on our microscope, and this is a picture of our microscope. I purposefully removed the detailed picture of our setup. We saw something like this, okay? And to an inexperienced observer, anybody would almost look at this and say, hey, this looks like cells, biological cells that you're trying to image. These are not materials, but this is how they look like. One important thing that I would like to remind you is that once I show you this image, we are actually illuminating this entire area in one shot. So it's a beam of light which is a diameter about 25 to 30 microns, all right? So we are not doing a confocal microscopy measurement where we excite a local point and scan and generate an image. We are exciting the entire thing, and we are taking a picture through a CCD camera or your cell phone camera or just what your eyes have of this entire area that light comes out of. Now, what was surprising is that if we looked at some of these crystals, we found a very funny thing happening. You see, this is one crystal. This entire thing is emissive. Edges are more emissive. But if you look at this spot over here or this guy over here, which is blown up over here, you suddenly see that it almost winks at you. It was looking at you and it suddenly closed its eyes, the light stops coming out, and if I plot the trajectory, you again start seeing this very abrupt changes in intensity, which randomly changes over time. One of this is for this, another one is for this, I don't remember. But the point is that this was perhaps the first example of a bulk crystal in which we could clearly observe blinking or blinking, or you can call it winking phenomena in these materials. But we were not the only people to see this, because this was a very interesting field. A lot of people were working. The only thing that we could tell is that this blinking process were perhaps related to something of metastable trap, a trap that exists for some time and then disappears after some time, and that can explain this kind of a blinking. And something similar was observed on this 200 nanometer by 30 by 30 nanometer cube crystals of methyl ammonium lead iodide by Ivan Sheblikin in Sweden, and they saw this very large intensity fluctuations as well. And their paper came out, I think, a few months before ours, but ours was a bigger, bulkier bulk system, theirs are not. And they explained their observations by a process which involves the diffusion of charge carriers in the system and also the diffusion of these quenchers. There is something which is a quencher that is there, which is a trap which quenches the emission, and that quencher can move around in the crystal one place to the other, and that can explain this kind of a phenomenon. So at that point of time, we thought that if really diffusion of charges or quenchers are involved, then why not we try out and look at other samples of other materials, which, for instance, we can have methyl ammonium lead bromides or Br2I or Bri2 and other things. And because these crystals, these materials essentially have been known or shown few years earlier to have very long electron and hole diffusion lengths and diffusivities. That was a paper that came out in Science in 2014. And I'm going to tell you our results on this particular system, because the others are more complicated, so I don't want to go into too much complications today, where we got something interesting. Now, I am a spectroscopist by training, and my students do most of the synthesis. So you can say that I am a synthetically challenged chemist. I do not know how it sounds, but that's who I am. But so I rely on my students to make the systems and my collaborators, and they tell me that it's very, very easy to make. You just mix up the precursors and spin cast them, heat it a little bit, and you can generate easily with these very nice crystals. And you can see they are individual crystals, they are fused crystals. The AFM tells you that they are about 250 nanometers in height, so there's no quantum confinement there. Very sharp absorption and emission spectra. And what we did was we took the samples and we put it on our microscope, illuminated the whole thing again, the whole thing together with a beam of light, and then these were bulk materials, and this is what we saw. It's a movie. This is, I'm showing here 5 different crystals which are spatially segregated from each other. This part here is, entire thing is a glass cover slip. This is a small area, of course, but if you look at them, they also seem to undergo this intensity fluctuations, right? Now this, if you look at this guy, this guy, this guy, this guy, you'll see that there is no correlation in the intensity fluctuation between these different crystals. And second, and that tells you something, that tells you that this fluctuation is not happening because of my laser intensity fluctuations or detector issues or any other thing, okay? Because they are very close to each other. And the second is that this fluctuation that you are seeing, they are not really happening from on-off kind of a thing. They are going from a bright to more bright or a dim to more dim kind of a fluctuation that you see. And if you look at the intensity fluctuations, to many this will look like noise, but it's not. Because if you take cesium lead bromide, this is the intensity fluctuation that you will observe. This is for the background. So what you see in the blue and the red or the maroon curves are actual real fluctuations. And these fluctuations which are abrupt fluctuations, high frequency fluctuations, is what we call as the flickering behavior of these microcrystals, flickering or blinking behaviors. Now there's something really much more interesting if you look at them closely. If you look at them, you'll see that within each crystal, a point here spatially separated by more than a micron over here or here or here, those guys, those regions seem to be fluctuating together. It's not that here and here intensity fluctuations are different. How can we tell that? You can take a crystal and you can plot the intensities at, let's say, 1, 2, 3, and 4 locations. And this fifth one in black is for the entire crystal. And if I look at them, I normalize the data and plot it, they almost look like identical to each other. So clearly, as a function of space and time, there's some sort of a correlation in the fluctuations that's happening in these crystals. They're behaving almost like I'm looking at a single nanocrystal or a single molecule, which is weird, which should not happen. And we have been investigating this phenomena over the last 4 years or so, and we have done several experiments, including looking at the blinking or the flickering behaviors as a function of size. And we found out, we actually figured out there's a way to quantify this spatial temporal correlation. And I don't want to go into the detail because there's a general crowd there, but we can generate some sort of a Pearson correlation or cross correlation coefficient number. The higher, closer to +1 it is, the better it is. And you can see this from an individual crystal, we can get a mean value of 0.97, which is really, really high. And if I plot it as a function of size, you can see all the stars are the correlation coefficients. They are very high. The mean value is like 0.9, okay, which is really 0.95 is the mean with a small standard deviation. So it seems that irrespective of size, within the limit of a diameter of, say, a half a micron to about 3 microns or so, you get this highly, highly correlated behavior in terms of this intensity fluctuations. Now, I'm going to come to why this may happen in a little bit. Now, another thing that we looked at was these crystals which are fused to each other. Now, unfortunately, in our lab, we don't have a setup where we can correlate SEM data with our spectroscopy data. So we have to do the experiments separately. And if you look at these crystals, they are fused. I can find another crystal in my optical image which are also fused. So I'm showing you the data for that. And if you look at this, you will see there are 4 grains, one here, one here, one here, and one here. And even these 4 grains, if you look at the intensity fluctuations, they seem to be happening together. This was really fascinating because this tells you that even if I take these isolated crystals, they would show this behavior, and if I fuse them, which is what you would do to make a film out of it for application purposes, even then you can see these kind of correlated fluctuations. Now, does this phenomena occur only for this disc-like structures? We investigated later on these rods because we wanted to show that this phenomena may actually be more general than we thought. And we looked at these rods of methylammonium lead bromide. Again, they are pretty big crystals. They are actually single crystals. They're much better quality crystals. And we saw that this is a rod which is approximately 1 micron. You can see it's a video. Its intensity is fluctuating. And if I look at 3 different locations in one edge, other end, one end, and in the middle, and if I plot the intensities, you again see such highly correlated behaviors. This can be quantified easily because since I have a rod, I can take a line along this direction and I can correlate the intensity trajectory that I have, that is basically intensity as a function of time. I can correlate each trajectory to another pixel's trajectory, and I can generate a map or a profile of the correlation coefficient as a function of the distance. And you can see it's very, very high. If I take bigger size rods, which is approximately, I think, 2 microns or so, we see that the correlation behaviors are there, they are predominantly there, but there are small little bit fluctuations that are there in terms of the relative changes. The events are happening, but the relative changes are not always as close to what we would have for a smaller crystal. And obviously, we will see a smaller value, relatively smaller value of the correlation coefficient, but still the numbers are very high. Now, if I go to a even bigger crystals, which is approximately 3 crystals, 3 microns in size, you start seeing weird behaviors where you can see that this side and this side seem to be fluctuating almost independently. However, there are instances that you can see, which I have marked here, where they seem to be also doing things together. I'm going to come back to that later if I have the time. Now, the question again is, how am I doing on time? I don't know. So, all right. I'll just let me know, 5-10 minutes ahead, okay? So the point is that how is this phenomena possible and why is this phenomena happening for such crystals, okay? So these are the 2 questions that I wanted to address before we move on, if there is time. Now, one thing that from the literature we know is that, and this was first proposed by a very well-known scientist, Paul Barbera, who worked on conducting polymers, and he showed that conducting polymers have many chromophores in it. So technically, it is not a single die. But even that conducting polymer with many chromophores in it, they can undergo these fluctuations. This is one of the first movies that or the data that I showed you today in the beginning of my lecture. And he explained this idea as there's many sides that are absorbing, but I can have transfer of this energy from different locations to a lowest point, which is undergoing this emission, and that is how he explained it. Essentially, this lowest point can essentially have some sort of a trap that can come in, and he attributes it to the presence of oxygen coming in and doing something and creating this defect, and that's how this quenching happens and this blinking happens in conducting polymers. And later, many other people have shown similar things, but Ivan Shablikin has also shown that this few 100 nanometer rods of perovskites, methylammonium lead iodides, they can generate this super traps or the super quenchers, which can quench a large number of electron holes or carriers through non-radiative processes. And he terms this as super traps. So the idea is that you need to have this metastable stable super traps or quenches that has to be present. And the word metastable is very important here. What that means is that first, these have to be induced by light and or the moisture or whatever it is. It is very difficult to characterize them because they're very, very few in number. What will happen if there's a large number of these traps? I will never see such arbitrary, abrupt fluctuations, because again, if I have a large number of traps, those points or those fluctuations will cancel out, and I will not see these sharp jumps that I would see here. Okay? So you need to have a few of them, and why they have to be metastable? Because sometimes they have to be active, sometimes they will not be active. That is the idea. When they're active, it will quench. When they're not active, it will not quench. Light will come out again. That is the basic idea. So what we thought was that, yes, this is distinctly a possibility that we have a few of these metastable quenches, which when generated will quench a certain amount of a percent of the carriers that are in the vicinity, and then it will reduce the intensity. And when it goes off, then again re-emission starts over the entire crystal and the intensity increases again at a different point of time. Now, we have done several experiments to show, and as well as not we also Ivan Shablikin and a few others, that these also should likely to be highly charged traps. Okay? They are charged species and they are transient nature, but they are charged species which induce blinking. Now, how do we think are these charged species are generated or they are annihilated? Because remember, they have to be metastable. For that, we did some interesting experiments where we actually put an entire chamber on top of our microscope. So this is the stage of our microscope. We have an inlet and outlet, and this is like a chamber that we have made where we could purge in any gases that we want. And by doing the experiment in the presence of nitrogen, oxygen, in the absence of water and the presence of water, we could show that there's some amount of moisture or oxygen is required to show this phenomena, to observe this phenomena, which tells you that these charged quenchers are triggered by the reversible adsorption or desorption of oxygen, moisture, and more importantly, light. Light is definitely doing something there, which we still don't understand exactly what. Now, this is kind of related again to the first system that I talked about, which were the nanocrystals. If you remember, it was dark and they were popping up one after the other, right? So this is somewhat similar, but in the reverse fashion. Here, moisture adsorption, desorption is leading to a quench state and or oxygen, moisture, whatever. I don't know what exactly the quench species is leading to this on or bright and the dim states. All right. The first thing is the existence of these metastable quenchers. The second thing is more intriguing because this size of this crystal is in the order of 3 microns. I am exciting the entire crystal simultaneously. So if I have to have a correlation in the intensity fluctuation here and here and here and here, then the carriers that are generated, the charged species that are generated upon photoexcitation, electrons and holes, the guys that are generated here, 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 and here, they need to talk to each other. It's like once you have a internet problem in your PC, first thing that you do is find whether the neighboring person sitting close to you or the next door neighbor also has the internet problem or not, right? Because then you know that you have to communicate, correct? So here also, if they have to recombine together at one point of time, species which generated very, very far away, they need to communicate with each other. So the question is, how do these photogenerated carriers communicate over microns length scale, which is actually a far more difficult question, I think, as compared to the previous point that I just talked to you about. Now, one way to think, chemist's way to think would be, okay, are these electrons or hole delocalized over this entire crystal, which is unlikely because these are semiconductor materials, but there is a possibility in certain states it is possible, but unlikely. Now, the other possibility is that there is a very fast, which we know from literature, that the electrons and holes are diffusing very, very fast in these systems. They are moving large distances, and typically the electron diffusion length is in the order of around 2 to 3 microns. Okay? And remember, beyond 2 to 3 microns, we do not observe such a correlated blinking phenomenon. Okay? So what we thought was that, all right, can we check somehow whether there is some evidence of excitation energy migration? That means if I generate excitation energy or carriers in a locally somewhere, can I figure out how to, a way to see whether they can migrate far away from here to here and then recombine or not? Okay, that was one of the questions that came to our mind. Of course, there are other possibilities such as waveguides or excited polaritrons generation. I don't want to go into the details. That's for another day perhaps. Now, one simple way to probe this long-range energy migration is to convert our existing setup. This is what we have. I'm going a little bit technical because I think it's interesting from the point of view of optics. When you do a measurement in a fluorescence microscope, you have to focus your light to the back focal plane of your objective lens. And that way, you essentially get a beam of light coming out from above you have a huge area of illumination. But we don't want that. What we want is to locally excite at a point that's typically done in a confocal microscopy experiment. So in a confocal microscopy, what you do is you locally excite things at one point, not over a large distance. And a simple way to do it is to just get rid of this focusing lens. And once you do that, you have this beam of light is coming in, and this beam of light, when it goes to an objective lens, it's a very high numerical aperture objective, you essentially focus the light into one point in your sample. Now, the emission that emerges from this point essentially is collected by the same objective lens and is detected with a camera like our cell phone cameras or our eyes. So we can basically generate an image straight away. So here's the idea. We modify our setup, and this is again, this was demonstrated in 2016. I think this idea was demonstrated by Van den Bout, is to focus the light to the objective lens, and imagine I have a rod. Then if excitation migrates beyond this excitation volume, and I can do this experiment because my rods are few microns, then I should be able to detect the emission from beyond the excitation volume. If I can see that, that gives us some evidence on excitation energy migration. And so this is the idea. You locally excite, you look at things far away, and we can remember that we are doing this under one condition, that the lifetime of the carriers are also high. That means if the carrier lifetime is very fast, then before they can run away and recombine far away, before they can run away, they will recombine. So I will never be able to actually visualize this movement or motion or the diffusion of the carriers beyond the diffraction limited spot. That is the idea. Again, this is the idea. We are focusing the light at one point. We are looking at the spread of this focal volume. This blue line here indicates the excitation profile of my laser. This green light here, if I see something like this, which is much wider than the blue line, then I have evidence for excitation energy migration. This is the result. This is my wide field excitation, where you can see the entire crystal. This here is the confocal excitation, where I locally excite over here. You can still see the entire crystal. If I take a line and make a line profile, you can see this is my excitation laser line, and this is what I would see from emerging, the emission that emerges out of the entire crystal. Same thing for the rods. If you look at the rods, this is what my rod is. If I excite here, which is shown locally, which is shown by this blue dot, I can still see emission emerging from far away from the local region. This tells us, and I'm going to skip the details here, and I'm going to skip the details here as well. This tells us that there is indeed the possibility of excitation energy migration in these systems through carrier diffusion processes following photoexcitation. So our model essentially involves three processes. One of the processes is the radiative recombination of carriers, where carriers are generated. How long is the time to recombine? Okay, that's in the order of 50 to 100 nanoseconds, but it can be longer. But there's also some sort of a carrier migration in the order of time scales in the order of 100 to 500 nanoseconds. That's longer. So while it's moving, it's also recombining. The other one is the formation and annihilation of quenchers, and that is the slowest step. And we all know as chemists that the slowest step is the rate determining step. It is the slowest step that is going to determine the time scales of my fluctuations in this particular situation. And therefore, what the idea is as follows. Once you generate these super traps or these super quenchers, these electrons or holes or these electron hole pairs or excitons, they basically sense the charge and they keep running from very far away because they are moving around very fast in the crystal, and a certain proportion of the entire crystal emission intensity goes down. It never goes down to zero level because it's a bulk material, so my emission is still dominated by the bulk, but a certain percent of those carriers get quenched together, and we see a depletion or enhancement of the emission intensity of the entire crystal. And this essentially explains the background that we see, on top of which we see this multi-level blinking. And also, this also explains this multi-level blinking because if I consider only a single quencher being present, I will basically have two intensity levels, but we don't see that. If we have many quenchers such as those that we are generating, then we will not see any fluctuations or not see any flickering or blinking. I'll have rolling undulations. We don't see that, which means that there are not very few of them, but neither there are many of those quenchers that are formed in each of these crystals. And we have some additional evidence for that as well, which I don't want to talk about today. How am I doing? Okay, absolutely great. Then I'm good. All right. So as I told you in the beginning that I'll try to touch upon something that as scientists we often encounter things that we don't understand. And I am very proud to always say that no, I don't understand, because that makes me think, that makes others think as well. So I'm going to share with you some of the results which are rare events, which are not commonly observed, but which are very bizarre and very strange. And this involves changes of behaviors as a function of time. Okay? When I say blinking, blinking itself is a function of time. Why? Blinking is nothing but intensity plotted against time, change of intensity with time. When I say correlated, a spatio-temporal correlation, I'm talking about here. Are they doing things together or are they not in sync? So if I say that's correlated, then this is uncorrelated. But when I say that temporal heterogeneity in this behavior, what I mean is that it is neither this nor is it random. That is changing as a function of the data acquisition time. That's giving an example of that. If you remember, I showed you this data where you could see that this part and this part most often does not undergo fluctuations synchronously. However, if you look at only this region and this region, they are undergoing simultaneous fluctuations. But once in a while, you will see that the entire crystal is undergoing a fluctuation altogether. Okay? I showed you before. Let me give you another example. These fused crystals, again, these two of them are fused. There's a barrier here. You don't see it, but there's supposed to be an energetic barrier here. And these are the profiles for 1 and 2. If you look at the green and the red curve, don't need to bother to look at the blue curve. And if you look carefully, you will see that for the first 10 seconds, they seem to undergo a correlated intensity fluctuation. However, in the next, beyond the 10-second time scale, you will see a completely different behavior. You see in the first 10 seconds, I am given you a few examples here between 0 to 2 seconds and 5 to 6.5 seconds. They're clearly correlated. This guy here, this grain and this grain. Beyond that, if you look, you see that correlation disappears. That means even within a single trajectory, I can have a situation that I have a correlated fluctuation happening for some time, and then it stops. There's no correlation anymore, and it can come back again, which was very weird, but we did have a hand-waving explanation to this. And the idea being that if I have these charges and carriers migrating, diffusing very fast in these materials, imagine that's what's happening here. It's going from one end to the other, this to this, so on and so forth. But suppose I generate a few quenchers in these interface regions, and these are charged quenchers. Let's say they're positively charged. And if I have positively charged, then electrons will get attracted and it will actually affect the barrier. And you can essentially change or create a barrier over here, and this barrier can stop the diffusion of these electrons, holes, and excitons from one grain to the other. And this is, again, a metastable process. Once this barrier is removed because of the removal of these quenchers or any other things, then it will go back to the situation which was the original situation. Or it may not get removed, and then you go from here to here to here, and you stay there. First it was correlated, then it became uncorrelated, the way I showed you. So one thing that I never, I've still not understood, this is actually published data, but we say that we don't understand, is the situation that you show here, 1, 2, 3, 3 grains that I want you to focus on. Again, these fused crystals. See what's happening. 0 to 4 second data, there's red curve, blue curve, no correlation between 1 and 2. 17.5 seconds to 20 seconds is a clear correlation. They are doing things in sync. They are fluctuating in sync. 16 to 70 seconds, what's going on? If one is going up, the other one is going down. Again, 29.2 to 30.1 going up, the other one is going down. This almost looks like what we call as energy transfer in a donor-acceptor system, where you have a donor and an acceptor, where energy gets migrated from or transferred from the donor to the acceptor. Intensity of one of them goes up, intensity of the other one goes down. But here I have these crystals. What is forcing the carriers to go from one of the grains to the other grain? That remains a mystery. We have still not figured out. I would welcome suggestions from the audience or anybody who's interested to propose anything. All right, so this is almost my last slide. So what I want to summarize, that what we showed is that by breaking some of the old notions, we showed that quantum confinement is not necessary. You don't need to have a single molecule or a single crystal to have flinking or flickering phenomena. We observe it even for crystals under special circumstances or special where they have special properties. So quantum confinement is not necessary for blinking, and entire bulk materials can also undergo blinking behavior. The second point that I would like to mention is that can this phenomenon be used for devices? Maybe. I don't know yet. I can imagine that if I have a long-range interaction or long-range communication, maybe it is going to be useful. Maybe we can use it to develop some sensors for detection of oxygen and water, because those are the ones that are behaving as quencher, which is resulting in the fluctuations that we call as blinking and flickering. And of course, there may be alternate explanations for this bizarre phenomena. What I have proposed to you is a phenomenological model based on our experiments, and not really a detailed theoretical analysis. No theoretical analysis actually has been invoked to explain what I have shown to you. I'd like to thank my students. I have got 2 acknowledgement slides. Most of the work that I talked to you about is done by Nitin and Tejamani. Nitin has graduated and doing a postdoc in Singapore, and Tejamani, who's here and here, he is about to graduate. He's a stellar graduate student. The rest of my group, you may remember Somnath Kashit, some of you. He did his PhD from here with Sion. He was a postdoc with me for about a year. And I'd like to thank my colleagues Anshuman Nag in ISR Pune for providing me samples of nanocrystals, and Shaibal Sharkar from Energy Sciences in IIT Bombay for also providing me with samples, and SCRB for DST grant that they gave to me for doing the confocal excitation, wide field imaging, and also MNRE Government of India for NCPRE. They helped me buy a detector and a microscope. And I'd like to also mention another person here, that is Ramya here, who has graduated long time back. She's the one who worked with Anshuman samples. The initial nanocrystals that I talked about was her work, and this guy Jaladhar and this guy, they helped me do a little bit of analysis on the correlations and so on. And again, thank you for your attention. It's an honor speaking to you guys.
So questions? Shan, are you?
Feel free to. I mean, anything and everything is okay. It's like
Yeah.
I see a lot of students here.
Thank you very much. I would like to know, could you try using Fourier transform or wavelet transform for analyzing your spectra, because that would tell you which are the prominent frequencies.
Yeah, we have done that actually. We have not done Fourier transform, but we have done wavelet transform on our data because the data is so noisy. And there is another component that I didn't talk about, is that there is a very slow modulation of the background intensity. So what we do is, in our analysis, we often do the wavelet transform and get rid of the trend signal, because we are not interested in the trend. We are interested in the noise of the wavelet transform, and we take that data out and we do the analysis. For certain types of certain systems, we have to do that, because or else we cannot do any analysis.
Okay, thank you.
Yeah, thank you. I didn't want to go into the details of all the techniques that,
Students? Questions? There must be a mic there.
Great talk. So these fluorophores, they are immobilized on glass cover slab.
the nanocrystals, if you or molecules, they are immobilized on glass cover slips. Yes.
I mean, that's just
So,
is it some specific.
So you can just
immobilization?
put them, you can just spin cast them on glass, evaporate the solvent, and the crystals are going to just get embedded on the glass. That's one way of doing it. Other one would be, way of doing it would be, you take the nanocrystals in a polymer and you spin cast the polymer. Then you also have the added advantage of oxygen has a little bit more difficulty accessing. Yeah.
My question was whether the immobilization will affect this photophysical behavior of the nanocrystals, and whether we can see the same thing in non-immobilized sample, like for example, keeping them in a vesicle.
Okay, this is a great question actually.
Freely move.
This is a great question. Thank you so much for this question. And this is a great question because no one actually knows exactly. Because the moment we are talking about a true single molecule experiment, where I am going to look at one molecule or one nanocrystal and collect the data over a long period of time, I have to immobilize it. I have no choice, because if it's in solution, it's going to diffuse out. I will not be able to collect the data. So what people have done is they have done fluorescence correlation spectroscopy kind of measurements, where a molecule or the crystal comes into the focal volume, undergoes some blinking or any other thing and goes away, and they have compared that data with what you have on a immobilized sample on a glass cover slip. That gives you two different time scales. But there is no way for me to tell whether the photophysics in the solid state, in the absence of solvent, in a glass or a polymer, is going to be the same as if it were inside a solvent environment.
So there are some studies by Gillard and group where they take giant vesicles and, I mean, inside that the fluorophore is immobilized.
Yes.
And that is
Yes.
That is not, so whether something like that.
Yes, I have not tried that experiment. So that those are typically done by people who are looking at protein folding or protein DNA interactions, and they make this giant it's called something like polymersome kind of a thing, right? And where they have a few hundred nanometers and they have it inside, right? No, so I have not done those experiments. For nanocrystals, I don't think anybody has done those experiments. They have been done for molecules, yes. Yes. But the goal, see, the goal of, their goal is very different actually. Their goal is not to look at blinking. Their goal is actually to look at some other processes. So, but that's a great question, because no one actually can say definitively whether this is the same or not. Yeah.
Arindam,
Yes.
great talk as usual. This is
Thank you.
really fascinating. Two questions, maybe rather naive. Is there a shape signature in the fluctuations? And the reason I'm asking is, let's say you have a single disc or two fused disc. It's the same material, same crystal, but you have a, so to say, a change in shape of,
Yes.
of the crystal. And if they're really fused, the boundary is the same material as well, unless there's
Yes.
a diffusional
Yes, the
gradient
boundary is the
and-
same
and
material.
so on,
Yes.
right?
Yes.
So the kind of bizarre data you saw with the fused crystals,
Yes.
I assume you don't see with the single crystal.
No, we don't.
And so is
We
there
don't.
something to do with the shape of the the
It is, so what you're saying is that the shape like this, they are connected together. That is, see, but we don't see this for every fused crystal. I told you this, right? That these are rare events. So maybe I will see this for 4%, 5% of the crystals, and students have to do the analysis. Only after doing the analysis, you can actually tell whether it is there or not. When you are actually collecting data, you can't even tell it. So it's only happening for a small fraction. The question is, why it is happening only for a small fraction and not for the rest of it? If you remember, I showed you a crystal which are 4 of them were fused together, right? 4 grains were fused together. So it doesn't happen nothing like this happens there. So the majority, even if I look at this guy here, they don't have a problem. It is only happening between 1 and 2. 1, 3, 1, 2, 3, no, not happen. So something
So it should
strange.
happen in a single crystal as well, a rare event. It doesn't have to be a fused structure. Right?
No, it can. So even for the rods, these are actually, my rods are actually single crystals, okay? These are polycrystalline, but these rods are single crystals, okay? So even in the single crystals, we see this. And we have a feeling that, well, we have evidence that there is a defects that is sitting here, which is actually quenching both this one and this one, but sometimes it is quenching this more effectively and sometimes it is quenching this more effectively, or maybe it is moving around a little bit. I didn't talk about that. I have a story, paper is being written up. So that may explain this kind of phenomenon. But this is actually a single crystal where, and we have evidence for it because when we do this local excitation, you are seeing this local excitation, right? You can see most of the time we see symmetric. Sometimes we see asymmetry in the intensity of the, why will that be? You can see that in one side I have more, on the side less. Then there must be a quencher sitting over here, and that is causing this part to go down sharply with respect to the other side. So if I scan my entire crystal by exciting here, everywhere else in different points, I'll be able to, in fact we have been able to identify that there is a different kind of defect, which is a more long-lived defect sitting there and quenching. So we know that in these crystals, sometimes, not every time, that there can be defects that are sitting there which is trying to quench, sometimes on this side and sometimes on this side, and maybe sometimes on both sides. That results in some of these rare events.
Yeah, so one just last
Yes,
question, follow-up, is there a material signature in this? I mean, if you
Excuse
have,
me?
is there a material signature in this? So if you have two such crystals of different materials,
No.
fused, why I'm coming to that is if you have perovskites, fused with silica or something like that, silicon or something like that. Do you
Yeah,
have,
so we see these crystals have been grown on the cover slip, silica, right? So all the crystals are grown on silica cover slip. So I wouldn't imagine that something special can happen in certain crystals. I cannot negate that possibility. But so far we have not explored that side yet. I have to think about this, and maybe I can change the substrate easily and see whether I have never thought about this. Thank you. I have to think about this. Yes.
Yeah. Good afternoon. So my question is regarding the light source. So this blinking phenomenon is observed only with laser radiation or other light sources as well?
We have only looked at it through lasers because our microscopy setup essentially is equipped with lasers. I would imagine similar effect because they are so bright. I would be able to look at get a similar phenomena even without lasers, provided I do certain checks in terms of the detection, because the intensity fluctuations that you see, that magnitude and amplitudes are going to go down if I use white light source. But because it's reasonably large, I think we should be able to see this even with intense band, not white light, but light sources which are not lasers.
Yeah. So just wondering whether this kind of flickering, winkering and all, winking, it happens in any biomolecules also?
Oh yes, biomolecules it does. And if you look at the fluorescent proteins, because remember that we have to have a fluorescent protein or a fluorescent molecule which will emit light, right? So for proteins like this, which have inherently a chromophore in this, I think it's called a beta barrel or something like that, where the chromophore is enclosed in this beta barrel, and the chromophore is actually a part of the protein or the part of the peptide. So this is a GFP. So when you excite GFP, there's a possibility that it can go to a dark state and it can stay there for a long time, and you can go from an on state to an off state which is blinking. In fact, since you mentioned this, I should tell the audience that this has a huge GFP fluorescence had a huge implication for what we now call as super resolution imaging, because it was the paper by W.E. Morner and Robert Dixon in 1997 Nature, in which they showed that this GFP molecule can be, not only can they undergo blinking, but using a 400 something nanometer light, you can convert an off state back to an on state. So you can have a control over whether the probability of the molecule, or control over the emissivity of the molecule, or the probability of emissivity of the molecule, and that property is used for super resolution imaging, called what we call as PALM or STORM. But yes, so that's the relevance of GFP. DNA molecules are non-emissive, so we can't do anything about it. So that's the most common form of biological chromophore which undergoes blinking. And it's also more like, it's not nice blinking like this. It's more like this. So.
Okay. So I have the last question, and
Sure.
I guess we should. So this time scale, and it's a very naive question. So there is this time scale of blinking will depend on the migration of these charges along with the lifetime of the trap.
I think it is being controlled by the lifetime of the trap,
More.
because that is the slowest. As long as the trap is active, I'll go in one state. But if the moment the trap is removed or the trap is inactive, I'll start having light coming out the normal way it's supposed to be. So you can look at it in either way.
So basically, this is the lifetime of the trap, then the time scale that you see in the
Yes.
flickering.
Yes.
And then you can change the environment of the trap, like you said, like you have oxygen, and
Yes.
and then the trap can be longer lived or shorter lived, and the flickering lifetime.
We have not seen that. We have not seen that. We have only looked at the frequency of blinking events. The reason is that when you look at a blinking trajectory in the ones that I showed you, you will notice that it's very difficult, as one of your colleagues pointed out here. We have to do a little bit of analysis to actually weave out the trend signal and we have to look at the fluctuations, that there's no clearly defined on and off state. So I cannot define that on time distribution, off time distribution from which I can obtain that, okay, if I change this, I can't do that. So I can only look at dwell time distribution. That means how long does intensity level pause at a certain intensity level.
Right.
Right. And if I do that, then I can, but the number of events that we get typically for is statistically not that reliable to infer a good lifetime, of the traps. Okay. But we have done another experiment, which I didn't tell you about, where you see, this is a data that is collected at 25 milliseconds per frame. So essentially, if something is happening faster than 30 milliseconds per frame, or 25, I don't remember. So then it would look like a jump, right? Big jump. Yeah. But suppose I do the same experiment with 1 millisecond, will it still look like a jump? No. Right. So what we found out that these jumps are okay till up to about 1 or 2 milliseconds. But if you go below that, they become steps, which tells you that that is really the time scale of something happening, the changes that are happening in this, the removal and the, we have not published it, we just done that experiment and kept it somewhere. But so lifetimes of the traps is different from the removal time or the absorption time, but we feel that that is the slowest time which is determining the fluctuations that we observe. Okay. So in that sense, it's different from the true sense, the true blue sense of blinking in nanocrystals molecules. So it's definitely different from that in that sense.
Okay. Yeah. Thanks, Arindam, and let's thank again.
Thank you everyone.
So we will help you to bring it down.
That's fine. You don't need this, no?
Oh. Okay, yeah. So you be there. I'll just request Ashish to give you the momentum. Yeah, at least. Yeah. Yes. So. Huh.
No, I'm telling you, it's not that bad.
Okay.
You have, I'm not hurting anything.
Okay, so I'll now request Ashish to present Arindam with the memento, which is as a token of appreciation. So here. Yeah. Any sequence. Yeah, need a signature.
Oh, I see. Oh, wow. Thank you so much. This is lovely. This is really lovely. My son and will really love it. We have, over this pandemic, we have started growing our garden in our veranda. So
these will go in. Yeah. So why don't you sit here? Yeah, because there will be a award ceremony. Yeah. So we will now move on to the next leg of the program. I will request Mr. Kadam to take care of the award ceremony. Mr. Kadam.
Good afternoon. This is my honor and privilege to conduct this award function. As you know, every year on such occasion, NCL Research Foundation recognizes the significant contribution made by our students, our staff, and our scientists who have made us proud through their work and their research. Before I proceed, I would like to briefly introduce NCL Research Foundation. NCL Research Foundation is a non-governmental body. It was created to encourage and reward the values and the culture of NCL, namely sustained excellence in science and technology, contribution in creation of wealth in society through science and technology, individual efforts beyond the call of normal duty, and constant endeavor to improve productivity and efficiency. With this brief introduction of NCL Research Foundation, I move on to announcement of the awards. We're handing over these awards in 5 different categories. Ladies and gentlemen, the first category of this award is Scientist of the Year Award. There are 2 awards in this category. The first award is NCL Research Foundation Scientist of the Year Award for the scientists in the age group above 40 years. This award is sponsored by Manekji and Srinibai Netwarwala Foundation. The award carries cash prize of rupees 40,000 and a certificate of commendation. Ladies and gentlemen, this year, NCL Research Foundation Scientist of the Year Award for the scientists in the age group above 40 years goes to Dr. Samir Chikali. Dr. Samir receives this award for Taylor single-site catalyst for the synthesis of disentangled ultra-high molecular polyethylene and functional polyolefins. Dr. Samir, if you could give your response to this. Tejas, mic.
Honorable Chief Guest, Director NCL, Dr. Ashish Lele, colleagues, friends, and my dear students, it is indeed a pleasure and an honor to receive this award. I would like to thank entire NCL family and NCL Research Foundation for appreciating my work in the area of olefin polymerization in general and functional olefin polymerization in particular. This area is little challenging area. You need to develop certain skills, and I think there is no other group in the country who is working in olefin polymerization and functional olefin polymerization. So I would really like to thank my students who have acquired these skills and who have contributed, which has made me to come here and accept this honor. I would like to dedicate this award to my student who have contributed for the area of functional olefin polymerization. I think such an appreciation also comes with some responsibility, and I hope that I will strive to meet the expectations of NCL family in the years to come. With that, I would like to thank all of you for patient listening. Thank you.
Thank you, Dr. Samir, and congratulations once again. The second award in this category, NCL Research Foundation Scientist of the Year Award for the scientists in the age group below 40 years. The award is sponsored by Dr. R.A. Mashalkar Endowment Fund. This award also carries cash prize of rupees 40,000 and a certificate of commendation. Ladies and gentlemen, this award goes to Dr. Rahul Bambure. Dr. Rahul receives this award for understanding in vitro and in vivo unfolding and refolding of therapeutic antibody fragments and monoclonal antibodies. Would you like to say something, Rahul?
I'm very humble and thankful for this award from the RF Foundation. So this particular work majorly involves understanding and unfolding and refolding of recombinant proteins. So most of the work performed by my past and present PhD students, and I'm really thankful for them for this hard work. I would also like to acknowledge the help from like CSIR-NCL and my industrial sponsors for this particular project, majorly Reliance and the Serum Institute of India. And also would like to acknowledge the help from my bioprocess engineering staff, Mr. Jagtap, Mr. Thakar, my family members, and my parents. Thank you. Thanks a lot. I appreciate it.
Congrats, Rahul. The next category of the award is NCL Research Foundation Sipla Hamid Award for the best process of the year. This year, we have 2 awards in this category. The award carries cash prize of ₹25,000 and a certificate of commendation. The first award in this category goes to Dr. Santosh Maske and Dr. Sanjay Kamble. I also request his team members, Dr. Mahesh Pisa, Ms. Saili Jagtap, Ms. Padmaja Fulewar, Dr. Sunil Bongle, and Dr. Sanjay Bhorikar to please join in. The team receives this award for the process development of a key step process for rosuvastatin. Let's have a big round of applause for them. Thank you, team. Congratulations once again. Moving to the second award in the category of NCL Research Foundation Sipla Hamid Award for the best process of the year. And the award goes to Dr. Amol Kulkarni. I also request the team members, Mr. Ranjit, Mr. Anil Palgadmal, Mr. Uday, Mr. Naneshwar, Dr. Ashutosh Ambade, and Dr. Samir Chikli to please join in. The team receives this award for the continuous process for the synthesis of hydroxy-terminated polybutadiene. Let's have a big round of applause for them once again.
First of all, I thank the NCL Research Foundation for selecting us for this particular award. This was a team effort between the 2 departments, 2 divisions, and quite some efforts have been put by students. Mainly, I thank the students who were involved in this because there was a lot of meticulous work that was done over the last few years, and it was tirelessly developed actually. And I particularly thank Dr. Vadgaonkar, who initiated this process, and then my team, starting Pagarma, Ranjit Apkalkar and Ganeshwar Mache, who put a lot of efforts for making this entire process come true. Now we are going for the next phase for this project, and we are going to put a pilot plan for this particular work. So thank you again for the NCL, Director NCL for this. Thank you.
Congratulations. Moving to the next category of the award. The next category of the award is NCL Research Foundation Award for New Initiatives taken by R&D Support System. The award carries cash prize of ₹25,000 and a certificate of commendation. This year, the award is shared jointly by a team of Mr. Amol Bhagat, Mrs. Pooja Kulkarni, Mr. Dayal Ram Saini, Mr. Ajit Malwade, Mr. A.V. Mahajan, and Advocate K.P. Anil Kumar. Request the team is come. The team receives this award for their outstanding contribution while dealing with an NCL land case and securing a landmark judgment in favor of NCL and CSIR. Yeah, you can take the award in here.
Thank you, NCL Research Foundation, for considering us for the award. I would like to take names of few of the people who have not been named here, specifically my seniors, my Samira Kulkarni, my ex-boss, and Vishwanath. Vishwanath has been very critical in this throughout the process. And last but not the least, our legal counsel, Advocate K.P. Anil Kumar, who has not been able to be here today due to there's some court matter in Bombay High Court today, sir. So thank you on behalf of all the team. Thank you, Research Foundation. Thank you, Director.
Thank you. Congratulations. Moving to the next category of the award. The next category of the award is NCL Research Foundation Individual Merit Award. This award carries cash prize of ₹10,000 and a certificate of commendation. We have 4 awards this year in the category of Individual Merit Award. And the first award goes to Mr. Gati Nayak. Mr. Gati received this award for prompt services for library maintenance, plagiarism check, getting publications from outside journals, and conducting SciFinder and Scopus workshops at regular intervals. The second award in the same category goes to Ms. Vaishali Suryavanshi. Ms. Vaishali received this award for smooth organization of PhD admission process and handling CSIR official activities for all NCL PhD students. Congratulations. The third award in this category goes to Mr. Srinivas Dev. Mr. Dev receives this award for sincere and dedicated services to CMC, being always available and extending support during odd hours and prompt maintenance of analytical instruments. The fourth award in this category goes to Mr. Mahendra Balmeki. Mr. Balmeki receives this award for his unflinching and dedicated services to run NCL canteen smoothly and ensuring refreshment up to late evenings. Thank you. Congratulations. Moving to the last category of the award. The last category of the award is NCL Research Foundation's Director Commendation Award. We have 2 awards this year, and the award carries cash prize of rupees 10,000 and a certificate of commendation. The first award of Director Commendation goes to Mr. Gopal Prasad. Mr. Gopal Prasad receives this award for his dedicated and consistent efforts in ensuring the washrooms in all buildings of the lab premises, hostels and staff quarters are kept functional all through the year. Last but not the least, the second award in category of Director Commendation Award goes to Mr. S. Mishra. Mr. Mishra received this award for his professional and consistent efforts in managing and providing seamless support in transportation activities. Congratulations, Mr. Thank you very much. I now request Ms. Pooja to continue the awards for the students. And congratulations all the awardees. And while we are changing the host, I request all the awardees to come forward for a group photo. Please, all the awardees to receive award, please come forward to have a group photo.
पूरा पूरा हुआ हाँ हम एक मिनट
research scholar working with Dr. T. Raza and CSIR-NCL will take you through the next set of awards for the student under various categories. And we are extremely honored to have such a great opportunity to announce the awardees on the occasion of National Science Day. The NCL Research Foundation encourages excellence in research by sponsoring awards to acknowledge the talent of research students in various categories. So without any further delay, the first category is the NCL-RF Best Research Scholar Award for the year 2022, which carries a citation and a cash prize of rupees 10,000 each. So the first award we have Mrs. Uma Dev Guptapu and Professor Ashwani Kumar Nangia Award for the Best Women Research Scholar in Interdisciplinary Research, and this award is jointly shared between Ms. Priyanka Kataria and Ms. Swarali Hirlekar. I would like to request Endowment Award for the Best Research Scholar, and it is given in 4 disciplines. So the first discipline we have Physical Chemistry and Material Science, and this goes to Mr. Akash D. Nidhankar. I like to request him please receive your award. I request to all the awardees, please sit in the front so that there will be no delay. In the next discipline, Chemical Sciences, the award goes to Miss Sonia Rani and Mr. Siddheshwar B. Ankhade. In the catalysis discipline, the award goes to Mr. Nitin B. Mahane. In the engineering sciences division, this award goes to Mr. Sagar Chaudhary.
Now the second category is the NCL-RF Best Research Papers published in the year 2022, which carries a citation and a cash prize of rupees 4,000 to 5,000 each. The first in the category is Dr. Rajappa Award for the Best Published Research Paper in the Organic Chemistry Division with the highest impact factor, and it is to Mr. Sairam Dhyaneshwar Veer for his work published in Angren Chemie International Edition. The next award is NCL-RF Nanai Award for the best paper published in the Organic Chemistry Division with second highest impact factor. It is being shared jointly between Deepesh M. Sharma for his work published in Catalysis Science and Technology and Vinita Yadav for her work published in Organic Letters. Next is the NCL-RF Gupta Pardeshi Sanainani Award for the best paper published in Biological Sciences goes to Vasudev S. Wag for his work published in Carbohydrate Polymers. Next award is Dr. Krishnan Award for the best paper published in Material Science Division, and it goes to B.P. Shivamurthy for his work published in ACS Applied Energy Materials. Next is Dr. Krishnan Award for the best paper published in Computational Sciences goes to Ms. Shweta Mehta for her work published in Applied Surface Science. Next award is Dr. Joshi, Dr. Shivram Award for the best paper published in the area Polymer Science and Organic Inorganic Hybrid Materials goes to Mr. Navnath Ramrav Katke for his work published in Journal of Material Chemistry C. Next award is Dr. B.D. Kulkarni Award for the best paper published in the Chemical Engineering or Technology Award goes to Dr. Praveen B. Patil for his work published in Journal of Environmental Management. Next award is Dr. Paul Ratnaswamy and Dr. Miss Sunita B. Kulkarni Award for the best paper published in the catalysis. It goes to Anand B. Sabade for his work published in Chemical Sciences. Next award is Dr. Patel Joy Award for the best paper published in physics. It goes to Palak Patel for her work published in the Journal of Chemical Sciences, Chemical Physics. Next award is Dr. Saurabh Pal Award for the best paper published in physical chemistry or chemical physics. Goes to Geeta Khabare for her work published in Journal of Material Chemistry A.
The third category is the NCL-RF Best PhD Thesis Award for the year 2022, which carries a citation and a cash prize of Rs 5,000 each. And the best NCL-RF Best PhD Thesis Award for the year 2022 goes to Dr. Praveen B. Patil and Dr. Gargi Guru. Dr. Praveen B. Patil completed his degree under Dr. Vinay Bhandari, and Dr. Gargi Guru has completed her degree under Dr. Sake Singh. And now we have the last and much awaited category, and guess is? Yes, the NCL Research Foundation Agni Mitra Memorial Award for the best poster, which is sponsored by the Student Academic Office under various disciplines. The poster exhibitions that held on 23rd and 24th of February received an overwhelming response, and this year we have received under 170 posters, and we have 33 winners. We at NCL, as a part of promoting the Rashtriya Bhasha Udyog, there was a call for Hindi posters, and we received around 30 posters. So a special appreciation mentions to all those students who have taken efforts their work in Hindi. So all the winners will receive a citation and a cash prize. Now we will call their name one by one. Please come and receive their awards. So first we have the Catalysis and Inorganic Chemistry Division. So the winner, there are 3 winners. So I will call one by one. Mr. Ajit Kumar and Miss Kranti Salgaonkar, Mr. Rohit Kumar.
Under Biochemical Division, we have 6 winners. It is Miss Sanjana Verma, Miss Aishwarya Davkar, Miss Shweta Deshpande, Mr. Abu Junaid Khan, Miss Raquel Samson, and Mr. Dhananjay Mudibatkar. Since Shweta Deshpande is not here.
In physical and material chemistry division, we have 6 winners. Miss Ashwani Verma, Miss Apurva, Mr. Manoj Patil, Miss Maria, Mr. Shivamurthy BP, Mr. Tushar Singh Verma. I repeat, Miss Ashwani Verma, Miss Apurva, Miss Maria, Shivamurthy, Tushar.
Tushar Singh Verma. Under CEP division, we have a couple of winners. Miss Divya Dikshit and Miss Samreen Shaikh.
Under organic chemistry division, we have 8 winners. Miss Ashwani, Mr. Mohit Kumar, Miss Nutan, Miss Neelu, Miss Sadhna, Miss Priyanka, Mr. Tejas Anil, Mr. Pinku, Miss Ashwani, Mr. Mohit Kumar,
Hindi Poster Awards for different divisions. First is Mr. Indrajit Chauhan, Ms. Varsha Mahadik, Shivamurthy, Purvi Purohit, and Divya Dikshit. chance. Thank you.
I can see already students are leaving. I just wanted to tell you something. I have observed that there was a large number of women recipients of the awards. And I think, and I hope you have, them throughout the, like they will go in the leaky pipeline and you will see them at the end of the journey as faculty of some institution. And with that, NCL should be more proactive probably to have women scientists in the faculty position. And now, thanks. This event wouldn't have been possible without help from many people. So, Mr. Sunil Patel, Dr. Nikhilesh Chadhav, Ms. Sonali, Shri Ganesh Mane, Mr. Jagta, Shri Avinash Devteli, Shri Sanjay Askar, Shri Mr. Javed, Mr. Mane, Mr. Yogesh Mahajan, Mr. Mishra, Mrs. Asha Shinde, Mr. Deepak Jori, Dr. Swati Chadha, and also Dr. Tejas Kaidankar and Mr. Sanjay, who has joined in the auditorium management part now, and he has played a big role, and you will see eventually we will have a much more modern auditorium system with him around. With that, thank you all for your presence.